等下就有声音，我现在开了。
没有，我是说那个
这台还没上去。
OK， 就点确认一下。第3题。有了。所以要有 Good morning. I hope all have recovered from last night's festive dinner. So I'm honored to have this opportunity to introduce this morning's plenary speaker, Dr. Peter Thomas. Dr. Thomas received his PhD in endocrinology from the University of Leicester in England. Following postdoctoral training in fish endocrine toxicology at Texas A&M University, joined the University of Texas Marine Science Institute of the University of Texas at Austin. He was promoted to full professor in 1990, and he also holds the prestigious HEB Endowed Chair in Marine Science. Dr. Thomas' primary research interest over the past 25 years has been on rapid non-genomic action of sex steroids in fish and in mammalian models. He has published more than 300 peer-reviewed papers and many book chapters. For his outstanding contribution in work, he has received many honors and awards that I don't have time to enumerate today. I don't want to take away your presentation time. So this morning, we are in for a special treat, and I welcome Dr. Thomas to do this presentation.
Well, thank you for that nice introduction. And I'd like to thank the organizers you and the organizing committee for asking me. I'm going to be talking about characteristics of membrane receptors. It's going to be a brief overview. And I'm going to just talk about the essential functions in one. Okay. So we know the classic steroid genomic mechanism of steroid action involving the movement of the steroids into the cell binding to transcription factors belonging to the nuclear receptor superfamily and the transport translocation response elements and the activation of genes and proteins. And this process is fairly slow, RNA and protein synthesis, and it's been known for some time that many actions of steroids are too rapid to be mediated by this mechanism. And there's evidence that accumulated that also steroid receptors can act with the cell membrane that cause fast intracellular responses, activating second response. This is classical mechanism. Many people call it non-genomic, but it often isn't always. It's not always pre-genomic, but it does always involve activation of the second response. Now, there's early evidence for this rapid steroid action to the cell membrane. It was done by people working with progesterone induction of outside maturation. So as early as 1969, Smith and Ecker showed that progesterone induction of oocyte maturation is not likely to be indicating that it's a non-genomic mechanism. And then in 71, Smith and Eck and also Masu and Mako show that injection of progesterone does not induce oocyte maturation. If you incubate the oocytes in progesterone, it does induce maturation. So it's clearly acting on the cell surface. And then James Mahler worked on this extensively, and he showed that progesterone inhibits adenylyl cyclase, resulting in cyclase. So he shows that progesterone before sex. And in 1982, he with Sadler showed that progesterone binding detected on oocytes by photography. So it suggests that there's a membrane progesterone receptor. We've also seen a similar situation. So progesterone induction of oocyte maturation showed again that cell surface mediated when you injected the MIS inside the oocyte, it doesn't induce oocyte maturation. But when you apply externally, oocyte maturation. We also showed that non-genomic MIS induction of oocyte maturation. And we also showed its acts. So this shows that oocyte maturation. This shows induction of and it's blocked by concussive toxin. And there's similar findings to be found in other plants, particularly so it suggests that the progestin action is mediated by membrane progestins. Now, two advances were made before we discovered the receptor, and it's what it's in 1986. My first graduate student, stressful period, finally identified. We knew it wasn't. This was 17, 21, 20 meters, 21, 500. And another major advance was by Ronaldo Castillo. He identified biochemically membrane progestins. This was the first time membrane receptor clearly characterized biochemical mediators. And what he showed was the binding characters very different from the nuclear progesterone receptor mediated relations that suggests that it's receptors. And for 25 years, the whole labs tried to invent membrane receptors successful. And this is the problem because it prevented recognition of this rapid mechanism of steroid action. But in the last 20 years, 3 novel 7, 8 receptors have been identified that mediate. So, first of all, on Zoom, here he is, research scientists identified membrane progesterone receptors. It's the same year as the human genome was discovered. So we're also identified everything else. Then 2 years later, we have shown that the G protein And about the same time later, another group, very close at the same thing. The two separate labs. Then in 2014, the postdoc in my lab discovered that membrane antigen receptor. And I'm going to be talking about all these three receptors today. The problem is when you find membrane steric receptor, this is not receptor for receptor that everybody's been looking for that mediates a non-action. People have this what it means study the nuclear receptors. There's one of the systems receptor. How do we show these receptors? So by fulfilling certain criteria. So several criteria to designate. First of all, the structure is difficult. See here, multiple secondly, it has to be present in some tissue. We know and critically, it has to have a binding so it has to have high affinity, it has to be saturated, it has to be displaceable. And of course, it has to have specificity consistent. But this is just a gene mutation. This is a maturation. And you can see that this binds much better separate competition and 20 beta S, which is separate. Another step is the first thing we need to see receptor binds 20 beta S. That's what specificity is consistent. Also, we needed to show that this receptor can express different expressions. So in the original paper, we looked at the expression of other factors, expression. All of them, they are. Another criteria, of course, is binding receptor. So I'm just showing a little bit of data here. So the proximity ligation analysis shows that NPR is close to the inhibitory gene protein, less than 50%. And consistent with the protein, when you add the maturation is down reduction, which is blocked when you activate the. Another criteria, of course, it has to be hormonally regulated in a manner that is consistent with it. So here is the increase in receptive NPRs to oocyte maturation, and it increases to its maximum and then decreases to. And then also by knocking it out, by in this case the medicines oligos, you can see that it blocks oocyte maturation. So on the basis of these initial criteria, a lot of studies have been done by us. And most of them have an MPR. I'm just showing the studies that have been done. About site maturation, sperm motility, just human objects, genome secretion, cell apoptosis, a lot of non-reproductive contribution, as well as many other. And importantly, it has a glowing cancer. So we looked at prolactin, breast, and researchers. And the thing is, though, with all these studies, but the micronuclear cell is still the structure. So even though there's all these serious diseases associated with expression of this disease, still don't understand structure of finding. And this is really desirable in developing specific drugs to treat these diseases, these cancers, and other diseases. And also, it was the first drug in our view, find criteria for its designation as a receptor. So there's 5 members of MPRs, and they're members of the progesterone and given receptor. Designation. So these are the MPRs. And there's 11 members of this family, and they're related to the family. They have a bacteria. Different CRs, even though, as I showed you, they have people in the literature. They are cheaper. And the thing is, structures are not the structure, the 3D structures are 1% or less. The one approach is if you have in the same family structures known as some families, structures, you can do homology. And this is an effective strategy. You model your reception based on the structure. And what happens is recently the X-ray structures and connected receptors. What we also realized was so many functioning amino acid residues are shared between these connected receptors. So they look very similar. So therefore, we built construction of homology, MPR alpha based on the structure and connected receptors. So the first thing is we want to know homology, just the receptors. And there's a prediction based on the positive insight. But really, we show in most membrane receptors. This shows the antigens. And what you find is positively charged residues are usually positive with greater insight. It shows you that in this case, the antigens. So this shows the electrostatic potential map MPR alpha. See this a negative part of the positive. What this part predicts the negative part extracellular. Positive. This just shows up the quality. And as you can see, there's many more. In fact, there's no positive charges. So based on the adiponectin 2 receptor X-ray structure. We built homology. Yankel did this. We built this homology. And it suggests the internal embryons intersect beta gamma delta. So this shows a binding pocket homology, binding pocket based on structure. And of course, we know nuclear progesterone receptor provides a binding. Of course, all scope receptors will function. And receptor 20 carbonyl is very important. And also, so we model our homology model. We notice that this is the progesterone there. We notice that there's a close H1 interaction with 6. So this suggests that this is critical. So how do we test this? Well, we tested the binding specificity of steroids, which have the carbonyl and alcohol, the oxygen steroids, to see whether there's any difference in binding. What we predict would be that the oxysteroids would not bind. Now, the 3-carbonyl, unlike the nuclear PR, the 3-carbonyl doesn't have any amino acid. So it suggests then that it's not required. And again, to test the specificity of this 3-carbonyl, we checked the 11 keto with deoxy group. And and here you can see other parts. For example, these are aliphatic aromatic amino acids. And here's one of them. For example, lady 106 has hydrophobic interaction. So we also muted these key residues. The first thing we got is but this is a thing of specificity. Here, displace the curve of progesterone. Here, then when you remove this 20 carbonyl, and then with some other serotonin, here you have this binding, which is really high binding. Then when you remove this carbon, so this suggests that the negative specificity is consistent. Well, what we found was when we decrease, this is the testosterone androgen. When you decrease, remove that 3-carbon. So it still is this is the androgen. Out. Just in the same time. The hydrogesterone binds well, and so it means that the 3-deoxysteroids are not binding, which is consistent. And this is important. It's the basis of designing specific, not actively design drugs And then the mutational analysis, as I said, mailing 1 to 6 of the binding is kind of just when you mutate that position to amino acids, absolutely increase by loose bindings, whereas you mutate so this is predicted that then gluten is polar when you make non-polar acid polar so the critical cells and and also one of them is seeker, which is long. It's really interesting because adequate active receptors so one so this thing maybe this is supported by now what about this expert? Does it find results in our model? So there's this new alpha prediction and it shows that actually if this but it's unlikely it's not reducing this so homology it's protein structures how the gap between sequences. It's new methods out there that potential to solve these protein structures. But now I want to move to start with regulation of so, as you know, surge and elite secretion reduces oxide maturation, and it does so causing a switch to the steroids. In response to that, ISOCCE graduate student in my lab, Margaret Case, did a lot of work on signal pathways and we will be showing you and in response to narration, increase the the activation receptor causes the alpha subunit to decrease the cyclase activity, synthesis, reduce PKAs, and then the beta gamma subunits activate the kinase AKT to increase phosphodiesterase, also in activation cycle. There's some other evidence about kinase in our body. It is activated. So in response to this activating these two pathways, they don't be too acceptable. But interestingly, estrogen that is also naturally prevented by endogenous estrogen. So, move the source of estrogen increase. The fact that it's activated the signaling pathway suggests it's mediated. This kind of talk about second discovered two labs. First of all, breast cancer cells. It's very high affinity, specific binding. And it is sensitive cancer. It's massive. It activates the stimulator. So it's increasing. And identified some nuclear central antagonists oxygen and G1. So different research systems. Many research groups have studied it. This shows broker saturation, scattering, setback, behavior, device. So this is evidence in zebrafish that estrogen inhibition of circulation. First of all, it's present. Estrogen inhibit spontaneous maturation. And also that specific and then also increases maturation. This shows the oocytes still. And they also reports increased psychic activity. This just shows the effects of IC, SCI, see, they mimic the effects of this. The estrogen then maintains risk of activation stimulation. And this just shows silence RNA results. This is GVPD. This is inhibited by estradiol. But when you knock out when you block this, so the inhibition reaction of each It also causes GBS syndrome. Now, this was first identified in breast cancer, feeding out active pathway EGF activation. This is also but it's maybe dual mechanism. So the physiological condition this is change in the state of so since so when is our this shows during GPD, the SRNA and system and this time knock it out talk about the membrane antigen receptor. So it was first identified in social cells in 2014. It has high affinity. It's a member of the ZIP9A family, the zinc transporters. They transport the zinc into the cells, the cytoplasm. And ZIP9 is the smallest member of the family, and it activates these increases apoptosis in early stage, decreases apoptosis in early stage, but increases in late stage. And these are ZIP9 cells. Androgen receptor cells. It also has opposite effects with apoptosis decreasing in early, increase in late. So it suggests these actions are safe, and they activate differently. So in early stage, ZIP9 activates inhibitory T protein, late stage cyclase. And these studies show, in fact, using a different method in early stage, GI is activated, late stage, and it has opposite effects of zinc. Early stage, it decreases uptake of zinc. Late stage, upregulates uptake of zinc. Opposite for cyclic AMP. Early stage, it inhibits T protein, decreases cyclotoxic. A late stage, increases. So it looks like a stage-dependent slower anti-apoptotic effect. Early stage, it acts inhibits T protein, downregulates cyclotoxic, after decrease of and in late stage post-late it does the opposite. So third membrane is non-responsive. So three receptors, haven't talked about it. Just the receptor component one, but that's supposed to. And there are other functions which are important in telogenesis, fertility, in knockout leadership, collaboration with Dr. Steve that it shows that this really affects fertility. And then ZIP9 as a port, in other words, the zinc part. So that's important for the treatment of development, fertility, larval survival, and this is showing that ZIP9 for the bronze pump. So third membrane receptors then mediate numerous essential. So the discovery of the identity of the third membrane receptor opened up new avenues that perturbated, non-classic, often non-genomic, always rapid action. A lot of progress was made, and they were involved in a lot of functions. The details are often clear and they're associated with many pathways.
Thank you, Dr. Thomas, for sharing your lifetime journey with us for the identification, discovery, and validation of membrane receptors. So, Dr. Thomas,
I think this is a paradigm shifting work, right? Of course, G protein coupled receptors started with coupling activating G proteins, right? And then later we found many of the classical GPCRs do not use G proteins. And here you have a set of receptors exactly the opposite topology of our classical GPCR, but activating G proteins. Again, you presented so much novel data, for example, GI to GS switching. Again, as you highlighted in the last slide, how these receptors can cover the G proteins, right? What are the sites? I wonder whether we can learn from those identified G protein coupling sites with GPCRs to see whether similar motifs are also here in your receptors. But again, I feel like when people discover that classical GPCRs are not using G proteins, but very highlighted by always those paradigm shift. What you're presenting is also the opposite paradigm shift.
This working, yeah. So yes, there's some evidence that they don't always activate GPCRs. I'm going on Nadar, who works with oocyte maturation in Xenopus, Qatar University, has some evidence they don't always activate GPCRs. Of course, GPCRs also are found to bind to the nuclear envelope, and they can act directly because non-signaling changes in cell function. So I think you're right. There's many things that need to be investigated. We early on we looked at C-terminal, and we did find some sites, but they're not the same signatures that are classically known in the adrenergic receptor. And interestingly, I showed that ZIP9 activates two different G proteins. So does MPR alpha, but in the fish sperm, it activates a G olfactory protein, the stimulating. Whereas every other tissue, human and fish, it activates. There's a lot of complexity.
Thank you. Very simple question. Sorry if I missed it. Can you comment on how ZIP9 switch back and forth between the GI and GS coupling? What is the cellular environment that's central?
Well, that's good. I mean, this is at two different stages of follicular development. So all we really know is that at a different stage, we know, of course, apoptosis varies with follicular development. But the mechanism by which comes G proteins, it looks likely that the abundance of the G proteins of these two different G proteins varies considerably. Then we make coupling.
Here. I mean, hi. It's great to hear all this progress. In the documentary, two.
Right. Yeah, well, the Dr. Zhu's also knocked out the MPRs. But yeah, the issue with the knockouts, though, I mean, they're not the gold standard, in my view, because if GPR is knocked out in the hypothalamus, then we know that GPR is important for regulation of gonadotropin secretion. So the site is not clear. But yes, there's dramatic changes, which was done by mainly done by the Young Su group, the GPR, showing there's lots of changes, fertility, metallogenesis inhibited. So quite dramatic changes in the ovarian structure. And the same with ZIP9. The structure of the cortical vesicles inside the oocyte is disrupted. Post-ovulatory follicle breakdown is disrupted, so apoptosis doesn't occur. So a lot of different changes that we see in those models, and I'll send you the papers. It's quite a lot of this.
We have this memories of it's not to
Oh, I see. Yeah, we've looked at this actually in a human endothelial cell model or vascular endothelial cell, and why the two receptors. And we've shown, at least in this model, that as I predicted, that the early events in anti-pre-atherosclerosis, migration, and adhesion are mediated by MPR alpha. And then after about 16 hours, the nuclear receptor is also involved. And then by 24 hours, the nuclear receptor is dominant. So it looks like, if you think about a response to a hormone, the initial response would predict would be through membrane receptor, which would start this process in the general response of the cell, the steroid. Then it would eventually lead to genomic responses. And these membrane receptors are not much genomic. Some people are pre-genomic, pre-endocrine bracket. That's do end up. And another thing is they can regulate each other. So in the human biometria, MPRs at the end of the term, biometria contract after that. The maximum effects of the nuclear receptor shut up by MPRs regulating their substance. Checked up.
So I think I would take the prerogative as a moderator by asking you the final question. I would like to know about the specificity. You showed that there is a reciprocal regulation of estrogen and progestin in an OM interaction. I was just wondering whether the downstream G protein signaling is the same or different. You said they are reciprocally regulated in the oocyte maturation.
Yes, and we haven't examined that because I didn't show you, but it also regulates the messenger. So it's a genomic. So it's causing a genomic effect. At least.
Okay,
we don't know the signaling part because ERK is activated, so we suspect.
Okay, because I was just wondering the specificity, because when you say estrogen with progesterone, I would think that they should go a little different pathway, but it seems to be, that's what I'm asking. Or they don't share that.
They go through very different pathways. The estrogen, G1, which is a specific agonist, activates, stimulates, and MPR alpha do the opposite. Down.
Any other questions? If you have more, you can always ask Dr. Thomas at the break. Thank you very much for a brilliant talk. So while he is getting the slides ready, I would like to have the pleasure of introducing the second speaker this morning. Dr. Wei Yan received his PhD from the University of Turku, Finland. That's it, right? So after his postdoctoral fellowship at the Baylor College of Medicine, Houston, he joined the Department of Physiology and Cell Biology at the University of Nevada, Reno School of Medicine as a professor. So he now is a senior investigator at Lundquist Institute of Medicine Innovation at Harvard UCLA Medical Center and a professor of medicine at David Geffen School of Medicine, UCLA. So his work focuses on the understanding of the cellular and molecular events on spermatogenesis. Welcome.
Well, thank you, Dr. Zhang. And also I've heard a lot of talks on fish and so I'm not going to talk about fish, but goal is to try to accept emissions restrictions person solve this very interesting problem. So I'm going to talk about a story that we under development for the past 15 years. And so it's really it's actually microRNA family, and so much effort finding the best. But I really think that in order to find a little bit and so hopefully I can work on these microRNA clusters on fish in the future. Anyway, so just give you some context on the start of this project. That we turn the clock back to year 2006. We remember 2006. That's like a so the way that we discovered microRNAs was to so basically you ask this RNA and there's like serum factor like a bacteria so we spent like a year colonies and we identified 225 microRNAs. And so when we map those microRNAs to the genome, and so we surprisingly find 30% of all the singular microRNAs that we discovered map to X chromosome. So that means X chromosomes each with the then before looking at their expression profile tissue, and so surprised by the fact that 40% of those singular microRNAs, they are like extinct microRNAs. They are preferentially exclusively expressed. So that means X chromosome contains a lot of microRNA coding genes, like the microRNA genes, and they are preferentially or exclusively expressed. So of course, you know, I work with great, that's really interesting. Now, when we examine the expression profile in spermatogenesis, you can see here, you know, spermatogenesis contains mitotic phase that really refers to the mitotic concentration of ammonia, and then they enter the meiosis as meiotic phase followed by apoptosis. So during meiotic phase, we know that all the studies they compare and then take the across except sex chromosomes. So you have X, Y, they only paired in a small region, and then the vast majority of the sequences they are not they don't pair. So what the X chromosome end up doing is that physically segregated other sex body. So that's like a phenomenon sex deactivation. So that means in the model phase that all the coding genes are shut down. And so that means you don't have any expression and during the model phase, and then some of the genes forever. Now when we examine the expression of that, this X microRNA during meiosis, we notice that the model actually they can escape biotic sex MST. So this is very important. That actually is really required to make sense. And then we recording this thing actually genetics, and then later on follow up fish confirm that the vaccine microRNAs are indeed escaping MSCI. So the reason why I think this is important is that think about it. So you have the X also vaccine microRNAs referential specific use cells. And then they're supposed to be shut down during meiosis, but they are not shut down. They're active. That means they must be doing something. Right? And so we continue to analyze these vaccine microRNAs. And so this is like about 50 different species. This kind of region where you have this microRNAs. And in terms of sequence similarities, she's there. And then, but if you look at different species here, and you're going to look at this X microRNA clusters, they are flanked, they are both by two coding genes. Two filaments associated with the fragile X syndrome. And so if you look at a cross species, and that's starting from fish, and so this microRNA cluster, you can locate it based upon the coding genes that are highly conserved. And so in the species, we find actually microRNAs. But once you have there, and then so they migrate to the excellent. And if you look at the different species in which microRNAs actually are evolving so fast, and as you look at, for example, here, mice, and so they're rodents, supposedly that you have two coding genes, just like in the region. Then you look at microRNAs actually they're quite a lot. 90 and this side was 2, this side and the rest. They look at the horse, it's like 34 of them, and then you have this side. So basically, as I said, so you have this actually microRNA clusters, they are exclusively expressed in instrumental and then during meiosis and meiosis, they are not shut down, they maintain their expression, meaning that they must be consistently important. And also you look at the evolution, see that each species actually they have different set of specific microRNAs. To me, this is super interesting because it tells that they are doing something that really is spermatogenesis and they are evolving so fast. So that's why this tree is this basically find this identify this microRNA. And so we did a little bit of evolution type of study trying to look at where they come from. And this microRNA is actually usually this is focusing on time and to identify that spectrum here 5 to 6 so basically it's meeting. And so the key thing is that so we call it family because you look at, as I said, zero footage. And so you can find approximately 5 is the different network. And so our conventional ways to predict and so again, when we started things back in 2006, remember that time is also prior to CRISPR-Cas. How do we generate so spring the so southern block and then the injection, chimera, all these things. Remember, it takes 20 years. So my first postdoc started work by knocking out asking. So basically, we took one microRNA from this sponsor, and another microRNA this sponsor. So it took me 2 years to generate 2 knockout, and it turned out the knockout actually. So basically, it's that you will not be able to read because you know, read my career and my eyes. And then I got the second postdoc. By that time, the CRISPR-Cas available. I said, you know what? Since the triple knockout, doesn't any phenotype, so this time let's do this. So as you can see here, we generate the single microRNA cluster knockout. So here, it's remove this cluster, which is 4 microRNA genes, 8 microRNA genes, right? And then we generate this another single knockout, this is here 465, and then we generate this double microRNA cluster knockout, and then we generate triple cluster knockout. It took about 2 years. So second postdoc name is Dan Oliver. So we got to the triple microRNA cluster knockout. They did one day in my dogs, all of these things. So, and I said, what are the people we should continue from? I said, no, I have to take the family and 2 days. You don't know how to so I can make so anyway, so we quit it and then we British business and really financial so if you're interested in British let me know. Then give us this stuff. But anyway, so I don't blame him. So for postdoc, for 3 years with me, all this knockout looking at the process. So we got a third postdoc and said, you know what, you continue to work on this thing, but I think we decided to find it this case. So the file came in, and so we built up this thing and start generating one as a full microRNA knockout. And then I said, okay, there are only 5 of them, so why don't we go ahead and do all of this? Because the Cas made it possible. This time it will be the entire 5. And then I said, we'll start analyzing the phenotypes because we now had on the so basically, so for example, we started with 2 microRNA clusters, and then we now have the third one, 2, and then we now have 4. So what happens is that when you make 2 bonds targeting, obviously off targeting effects. So say that it affects them to become something else. Of course, you can bring about really get rid of that, but it takes longer time. I said, okay, this time design not RNA to remove this 5 at 1 so we needed so just 12 years and 3 postdocs and 2 quit 3 years and actually persisted and so finally we got something here. So, okay, all right. So okay. So what happens is so I list up here is the single knockout, right? Single knockout, triple knockout. As you can see, before you reach triple knockout, so the first thing you do is test the opportunity, right? So before, so basically, before you knock out 4 micron monsters, okay, but when you knock out 4 or 5 services, but if you look at other things like either links, fonts, utility, everything. So that's sort of like a really mysterious, but that's still thing that is very important. So one thing is that once we got this kind of data, and then the postdocs actually find what they don't want to touch it. But so we put the project aside for a couple of years until I went to different meetings, talked to some senior colleagues there, and then one of my colleagues actually he said, "Have you tested sperm competition?" My training is that when we evaluate fertility in mice and they get age and then 20 days later get pups, they're fertile. Is what we're talking about. He's saying that wow. So the rodents actually do this mating. So usually the female actually makes very fast. And then what happens is that you have the sperm actually end up in the female tracks. So you have sperm competition thing. And so he also mentioned that you think about it, it's logically you're not proceeding from that view. The reason is that these microRNAs, they are highly, highly variable. Like among different species, people within rodents, they still differ a lot in terms of sequence, in terms of composition of microRNAs. So they must be controlling something not like highly conserved. We know that spermatogenesis is highly conserved species. So if these microRNAs are so important, you will change the fundamental process in spermatogenesis. So probably even they have some effects, but in the subtle. So the phenotype you see so subtle, that's important. So that's I got encouraged and came back saying that you guys set up a meeting, go there and then watch their meeting as soon as they made, take the first meal out, put the second meal, and then my student came back saying that hey, I cannot do it. So and I realized that it's impossible. Watch there 30 minutes, you check out, and then the mice is really like annoying. So that's not the right way of doing that. So fortunately, I got help from my colleagues that he's saying that even with the population, so after PNST, and then we get HCG, usually give them like 8 p.m. So and then we put in the first meals, and then so let's go morning, 6 a.m. And so you check the clock, if the clock, you put in the second one. You're supposed to the operation peaks around 7:30, and then so by 4 p.m. so they second and then eventually you can have like at least 2 sequential meetings just by so I never heard about that. I felt like the function rule part of meetings and the second meeting actually in reality that's so we noticed that it's the first meal clock and then the second meal back there later on going to find the clock actually before the second clock. The reason how do I know that? Because I use the market clock and then so after 2 hours you find the second clock is fresh and then the mark that clock actually inside the page. They're suggesting that it comes out. I don't know if you get it out or it falls out. So don't trust the test. Sometimes they don't choose. So as you can see here, and so we first put in IPNG, that is like the person's metabolites normally sperm red and this will see here in this the embryos is red, this will definitely find the so if you put IPNG meeting first, and then in the morning, the second one, that's this 5 And so 100% the offspring are derived. So in other words, these guys have a chance they make first. And then we reverse that. So we do this knockout first, maybe, and then later on, like in the morning, second gene that is supposedly this is normal. It's like as competitive as the benefit gene, you're supposed to see 100%. Actually, it's not 100%. It's about 90%. You still have like a 10% gene offspring. So suggesting that this is like at least it's not as competitive as the gene sperm. And as I told you, this is not the real natural setting because we saw like it's like there's 6-8 hours apart between two mating, and so that's not really natural. So naturally in the wild, the between these two matings about. So what should we do? And I was thought about this thing. So why do we do the IVF? So in this way, we can mix the MPMG sperm with the sperm, different ratios, and then we transfer the embryos and see which one actually stagnate ratio between the MPMG and the sperm versus the knockout. By the way, so all the data suggests that they are less competitive. Of course, we need this artificial examination. Mixed sperm so all the data suggests that the sperm are less competitive. So they are the white sperm. Anyway, so I try to so of course the people see the competition. There are physical features, the fact that sperm they need to put together the fertilization, and the middle piece which enters the difference. So of course we did all this profiling, try to identify what happened. So because I'm running out of time, and so you have to trust my words. But the bottom line is we find that this my part is the complexity, regulating ability to transition efficiency. So, but instead they're targeting all genes. In other words, the higher species use spermatogenesis have like the normal genes that need to be targeted by the same set of RNAs, and so it's not because the increase protein size. But anyway, so I just want to wrap up here, and so in conclusion, we think that this microRNA family, they are regulating, it's like a fine-tuning spermatogenesis by regulating how basic spermatogenesis, so making sperm more competitive. Right? So finally, I want to acknowledge how your thought is still working. And I want to take this opportunity to President Day finishing my term in the energy March, and I took my goal to extend next year.
Thank you for presenting your very, very interesting questions. Yes.
Very interesting. How we miss. Have you checked the motility in the difference in the knockout? Check with the white.
Yes. So we have, this remember motility is underlies by CASA. And CASA is favorite programs by a high school, which is completely ridiculous to me. So it doesn't tell me what needs to be measured. Except it's many.
Please.
That's what I do. Yeah. Yeah. So, yes, yes. So I didn't have time to so, basically we identified the target and so basically I species. So we believe that sequence target is similarities and then you see how many is that. And then you look at that and so we did a whole lot of analysis to try to see same set of genes because they become more complicated and in the higher species, therefore actually that's not the case. So as I mentioned, that's because humans, huge genes that so they target conserve genes. Sort of that. Based on the RNA based on this systems. Then those genes actually are extremely expressed in the lungs. It's not like a system. Yes, because actually that one slide I didn't mention that. So we did a smart sequencing. So it's like total 15 then we have so this is the RNA sequence data. And also this the other this so they watch each other's exactly. So that doesn't mean they're not very important because you have to solve them. Saying that because I really think that my dream is these things. And because they have this hierarchy. The king and I use something there. And then of course fish another thing. So that it's all also it's not like seeing fish for sure. But we need somebody who really fish. So you guys should know storage of so we need an excellent. So if we can have to show this thing actually involve species, as I said, evolution later. That's the
So you mentioned that the immunobiosis increase the copy number through the process. Right? So does that mean all the mice will have more immunobiosis compared to the young mice? And how does the gene process being activated? How is it regulated?
And so I think that they are sort of like it's an adaptive response and somehow transform because features up there at the same time they can diversify by changing so the bottom line is this especially nutrition is a widely used mechanism for nutrition in this stage. Phylogenetic analysis. Do you know this cluster actually exists in the Drosophila? Right, amongst the Drosophila, there's the sperm storage. There's a lot of sperm competitions, and very easy to do the analysis. Yeah, so if you can help me to put that, my gut feeling is that there must be some primary similar. They are
Yes.
Of course, a lot of species pair bond for life. It'd be interesting to see whether they have the same patterns of microRNA or as much microRNA in species where there's no sperm competition, at least between individuals. So yes.
so that's
Ask the last question. So what's the stability of the members of this family of microRNA, and are the functions regulated by the stability?
so microRNA is the target. So right now we're just trying to see which genes are the matter of fact that they're found. The people message is that microRNA is the existing biological and they never work. They always work as a group. And so any paper you see a different outcome, you see bad phenotype. It's very rare case, but that's why if you check all these fake papers, they are rejected. Most of them actually deal with microRNA. So as I said, we now have 19. You see this really, so this is not really rate. Even the last they are the same thing. So they have a very subtle effects on the gene expression. That's why
Thank you very much for your beautiful presentation. Now I'm going to transfer the mic to
Yeah, although we are a little bit delayed, so let's start. Yeah. So the next speaker will be Professor Gong Hongyi from the local institute here, National Taiwan University. Actually, I knew him for a very long time, ever since he was a graduate student. So he specializes in fish genesis, yes, and then also fish biology, fish reproduction. So today he's going to talk to us about his very interesting work on angelfish and then the function of dead end 1 in the maintenance of primordial germ cell and sex termination.
Thank you, sir, Dr. Zhong's introduction. First, I would like to thank the open committees to invite me to give me the opportunity to present our recent works, especially in the end works, and especially thanks the President Qingfeng Zhang asked me to do the report. These are the PhD students, my peer student, co-advisor, Professor Zhang's making use of works. Today, I would like to talk the dead end. It's essential for the maintenance of primordial germ cells and the sex termination in the non-model fish, freshwater angelfish. As we know, that aquaculture is becoming more and more vital. The current projects shortage in the aquatic food availability as the human population is growing. The seafood consumption shifted from the fishery harvest to the agriculture. So to maximize the food production and minimize the ecological impact, the reproduction study is more important. But not only the edible fish, the ornamental fish is also an important industry in the aquaculture. And as we know that both selective breeding and the transgenic technology or even the gene editing is very important and high potential for improving the development of aquaculture industry. As you see, just like the GIP project, the genetic improvement from tilapias, and also the transgenic aquaculture salmons, and including the ornamental fish from the selective breeding or even the transgenic fish, just like the fluorescent dipper fish. And today I want to talk the angelfish. This all from the genetic improvement the technology. If this fish escape from the aquaculture containment, it possesses a potential to risk to ecosystem and environment. So how to solve these problems? We want to use the genetic infertility strategy. Farming the infertility fish is the most effective way to avoid the economic risk of the raising the transgenic fish or even the aquatic fish into the aquatic environment. It also shows supporting for the development of environmental response agriculture. Now we want to the strategy we want to target the genes that's essential for the primordial germ cells development. What are primordial germ cells in the telos? It's the primordial progenitor cells of the gamete that is specified early in the development by induction or by the inheritance of maternal provide determinant, including some maternal RNA and the maternal proteins. And this PGC they will form early in the development and migrate into the general region. In the deep system, the PGC are well studied. The PGC development has been extensive studies in several model cases, including not only the deep fish but also the Drosophila silicon, Genopus, and the Metacarp. And in these models, the PGC are autonomous performed by maternal surprise gene plays composed of many of the RNA binding protein and the maternal messenger RNA. And why we choose the dead end? Because the dead end is a vertebrate specific genes encoding the RNA binding protein was first identified in the deeper fish and explicitly expressed in the gene line. And then play a major role in the gene line development. And then was sure that it will protect the specific messenger RNA just like the downstream the VASA from the microRNA, such as the microRNA 430s, mainly this kind of microRNA mating inhibition within the gene lines. The loss of the ends will result in the primordial germ cell loss the ability to migrate activity and the mismigration, followed by apoptosis and the transdifferentiation, but with no effect on the development of somatic cell in the developing system. And how about the infertility control of transgenic ornamental fish? Because in last years, it was reported that the transgenic growing the fish appears to be striving after escape from the fish farms into the Brazilian streams and may threaten the local biodiversity. And in my laboratories, we have established transgenic fluorescent dipper fish and the angelfish lines, expression the coral, green and red fluorescent proteins by using the dipper fish CKMB muscle specific promoter enhancer. And in this year's my laboratory, we published papers with doing the toxic immunogenesis of their genes in the wild-type dipper fish and also the transgenic fluorescent dipper fish by using the CRISPR-Cas9 technologies, result in the all males and homozygous mutant dipper fish with a normal tertiary behavior without any spread to fertilize the sperm eggs of females wild-type dipper fish. So make it become effective infertility control technology. This then now by CRISPR-Cas9 can be successfully applied to the infertility control of transgenic fluorescent dipper fish to prevent the escape into the wild fish. Okay, and as they show the data, you can see the in the homologous mutants, the heterologous mutants, it will show in the males, it will show the normal tested development. but in homozygous mutants, you will see the testes are all repressed by the adipose tissue, and but this male fish still can maintain with the female white fish to make white fish to lay the eggs, but all the eggs did not be fertilized. So after 10 hours post fertilization, you will see that all the eggs will be dead. So make it become the effective fertility control technology. And the 10 years goes from the 2013, we made the transgenic pink angelfish using the debugfish muscle sleeve enhancer promoters to express the equatorial red fluorescent protein. Up to now, have been inherited to 10 generations. It's a beautiful fish. We want to search for a GMO infertility controls technology in this kind of the transgenic pink fish, especially whether we can establish a non-GMO infertility control. And why the angelfish? As we know, the global ornamental fish market size was estimated at 5.8 billion US dollars in 2022, and it is expected to expand at a compound annual growth rate to the 8.5% from this year to the 2030. Okay, and for the demand for the consumer and to enhance the ornamental fish industry competitivity, the new variety development is necessary. And why we choose a freshwater angelfish? Because it's one of the most popular water fish species in the aquarium trade worldwide. And as I tell you, we established a beautiful transgenic fluorescent pink angelfish lines, and but it has a large potential business opportunities in the ornamental fish industry. Unfortunately, the fluorescent fish species are not currently available for sales due to the GMO regulations in Taiwan. And the to develop the effective practical fertility control technology are important for this fish to the market. The specific aim of our study is to investigate the role of the dead end genes in the freshwater angelfish and develop effective fertility control techniques for angelfish. Especially, we will focus on understand the significance of germ cells and the angelfish. And from the cloning of the angelfish dead end genes and doing the expression analysis, we found its maternal supply by the oocyte, so you can detection its messenger RNA at its earliest previous stage. And we also use the hormone in situ to detection the messenger RNA expression. Especially, you can see that in the 2-cell stage, the messenger RNA can be detected in the 2 cells cleavage layer, just like the basals in the basals and the end in the debugfish. And in the adult, their expression is exclusively expressed in the gonad tissues, especially for in the ovaries, they are abundant expressed, more abundant, strong expressed than the testes, about 3/4. And by establishing the dead end now angelfish using the CRISPR-Cas9 technology, we can see the gene structure of the dead end gene in the angelfish, just like the other telos. They compose of the 6 exons, and in the exon 3, a very important allele motif is located in exon 3. So we designed the guide RNA target the exon 2. And from the angelfish, a pair angelfish, they can female can lay the eggs at the tubes surface, and the females will have the sperms to make it fertilize. Then using the 1-cell stage, we can co-injection the guide RNA and the Cas9 messenger RNA into the 1-cell stage eggs. And using the T7 ones assay, you can see the target size. The target size are effectively cleavage by the exonuclease. And doing the sequencing, we can see the from the F0, the somatic cells analysis, we can see that some deletion, different deletion can target the end genes. And how about the F1 generations? We use this F0 homozygote mutant, the F0 mosaic mutants to cross with the wild type females, then gather F1's offspring. Okay. Then doing the genotyping, you can see using the PCRs, you can see that you can see this is the heterozygote mutant and the homozygote mutant, and the mutation efficiency is about 50%. And using the F0's heterozygotes, we can cross together to the homozygote. And also the catapins to do the genotyping, you will see that there's wild type, this heterozygote, and this homozygote. And the ratios for the after hard work of my PhD student, the two patients, they identify 60 or 136 fish. You can get the difference, the three genotype. This matches the Mendelian inheritance 1 to 2 to 1 ratio. And from the we find the male bias development by the sex ratio, analysis based on the external sexual characteristics. In the wild type and also the heterozygote mutants, they have about the male to females 1 to 1 ratios. You can see the especially for the external characteristics, the male fish will have the raised with the raised head. And in the homozygote mutants, we also will find the 70% fish, they will have the raised head. These are male characteristics. But some of fish, about 30%, we cannot identify whether it's male or female. Especially for this fish, they did not have the springs in the gonads. In the genital pores, it's also hard to identify the male and females. But for their behavior, courtship behavior, this water fish are not out, the homozygote not out of the dead end. And we're using the females, this golden angelfish, to making with this they are now male fish. And you can see the females can lay eggs, and the males also will courtship with the females, and they try to give the sperm to get the fertilized, but it fails because it did not have any sperm to make it the oocyte to be the fertilized eggs. So after the 10 hours, after the 20 hours, you can see they are still not developed. And after the 20 hours post sperm, they becomes dead. And finally, all the unfortunate eggs will undergo the cell death in 24 hours. And from the morphological analysis of the gonad tissues, we cannot see cannot easy to identify this with 40 to 30% the sex undetermined fish. But from the dissection, you can see you cannot see the obviously obviously the wild type of heterozygote mutant their testes the structures the full of the springs is only the one empty tube. And during the histological sections, you can see in the female the wild type and the heterozygote mutants in the stems, you can see the mature oocyte and the pathogenic oocyte. And in the males, the wild type males and the heterozygote dead males, mutants, you also can see the spermagonia and the spermatic. So it did not see the much difference between the heterozygote mutants with the wild type. But how about the homozygote mutants? You can see we classify into the two groups of the heterozygote mutants. One group is they have the male characteristics, raised head, but another group is sex undetermined. But you can see that all this kind of two groups, they and homozygote mutants only have the one fuse for the gonad. And during the section, you can see the tested structures, but with no any springs inside this epigonal. Furthermore, we doing the transcriptome analysis to do the global gene expression profiles in the wild type, the male and the female, so using the ovary in the test and the heterozygous ovary in the test. And the hormonal mutants using the two groups. The group one is they have the male characteristics, and this group two is they cannot sex determine. Only these two groups they only have the empty the tested tube. And you can see from the PC analysis we can see the hormonal mutant. They are the genetic profile is near the wild type male and the heterozygous male, and the wild type ovary and the heterozygous ovaries are classical together. So from the whole transcriptome analysis, we can propose ours that and hormonal mutants. It seems it's very profile similar to the males. The males and fish, no matter what type of the heterozygous mutant, and especially we're doing the quantitative RT-PCR to analyze some sex-related genes, and for this males bias genes, this BMP15 and the GDF9 and the nano3, okay, they are female bias genes, and these VASAs and the DAZOs, they are also expressed in the germ cells. But after the hormonal mutants, all the genes you cannot detect it because the loss of germ cells. So this 5 germ cell specific gene cannot be detected in the hormonal mutant. And in the somatic sex-related genes, the male bias GSDF and the AMH, you can see the GSDF expression is similar in hormonal mutant, is similar to the wild type male and the wild type heterozygous mutant. And especially for the AMH, we can see the dose-dependent increase for the AMH. In hormonal mutant, it's a significant increase than the wild type testis. And the SUCCE also showing the similar pattern with the testis in wild type and the heterozygous mutant. So the mass specific gene expression is high in the bone of the and the mutant, hormonal mutant angelfish. And although the AMH is highly expressed, but it's much to our surprise, we see another important DMRT1, the male determination genes are slightly being inhibited. And in contrast, the FOXL2 are being slightly increased. And the female bias, the gonad or emeritus CYP 19A1A genes expressed in the gonad, and the CYP 19A1B is expressed in the brain. Those two, especially for the brands, they have been downregulated, similar to the low expression, similar to the male fish. And but although the gonad and metis genes are slightly increased compared with the testis, but it seems another has a significant difference. And we also checked the serum testosterone, the labels for the estradiol and the testosterone. And it's interesting the estradiol did not have the significant difference within the different 3 types and the genotype. But in the testosterone, we see the significant difference. Even in hormonal mutants are higher than the female. So we hypothesize that the must play not only a significant role in the migration, the cell maintenance of primordial germ cell, just like in the GBA feature, but also act as a germ cell key regulator to regulate AMH gene expression level in the gonadosomatic cells to be involved in the sex determination of the freshwater angelfish. Because in the dead now, we see the strong induction of the AMH expression in the somatic cells. And this strong expression of AMH will feedback the inhibition of the DMRT1 expression. And the FOXL2 will slightly increase. But in the CYP19A, A1A, and especially for the A1B, 19 A1Bs that are similar to the male fish. So this kind of strong express. So in what type of angelfish, as we told you, the dead expression is much higher than in the ovary than the testes. So the maintaining the ovary cell phase, we propose the dead end through the inhibition of AMH and cooperate. And as we know, the DMRT1 we also will inhibit the induced AMH expression and cooperate in the ovaries, the ductal tubes will cooperate with this dead end to maintain the female cell phase. But in the males, the dead end expression level is low, and the AMH expression level is high. So to cooperate with the DMRT1, they will make the maintenance of the testis cell phase. Okay. So behind the germ cells, no germ cell in the dead end hormonal mutants, we also observe the proliferation, the connective in the hormonal mutants using the stainings, and also to see the expression of collagen 1A, 1A1B and 1A2 expression. And interesting to also see the energy translocation. They did not supporting the germ cells formation, but the accumulated visceral fat tissues are increased in the abdomen, the dead end hormonal mutant. So we summarize that we first established this infertile dead end mutated endofish with empty gonal structure without germ cells by CRISPR-Cas9 the genome editing. Additionally, we observed the male bias phenomenon in the dead end hormonal mutant groups with most individuals exhibiting external male characteristics and normal sexual behavior. First of all, we observed the significant higher AMH gene expression in the dead end hormonal mutant compared with wild-type male angelfish and higher serum testosterone level compared with the female angelfish. All studies demonstrate that the presence of the germ cell is essential for the female sex determination in freshwater angelfish, which hypothesizes that they not only play a crucial role in the maintenance of PGC and the germ cell, but also act as a key regulator of germ cell and repress the AMH gene expression in the gonal somatic cell to determine and maintain the female sex. Okay, and we also doing the transgenic and PGC germline specific for reason. And for resident angelfish, we can tracing the PGC migration process up to the post-hatching, 4 days post-hatching. And this cartoons you can see this PGC will appear, especially from the once all the egg fish, then we will see the PGC, the migration pattern from the epibory and to the somatogenesis. And up to fourth, and it will migrate to the anteriors and the back move to the posteriors. And the finally is not as the jibbafish, the then will arrive the migration arrive to the general reach at only the 24 hours post fertilization. But in the angelfish, the PGC will continue to develop, and after the hatching, the 4 day after hatching, they can arrive to the destination. Okay. So I would like to thank especially my PhD student, Zhu Huijun. He gave this very wonderful result, and the grant supported by the National Science and Technology Council. Thank you for the attention.
Wow. So any question?
放在正中间，好可怕。
台湾的
哦，真的，我们看到
This is a very good idea. That's why we establish this germinalized concentrated fluorescent angelfish, and because we're using the especially I didn't mention about that we're using the non-tilapia promoter. It's also the secret fish, the angelfish, and the both are 4 kb promoter, and combined with another 1 kb 3.2 TR to make a very strong fluorescent angelfish. We can see this PGC from the only from the stereo microscope, fluorescent stereo microscope, can see the PGC fluorescence. Not you cannot using the confocal microscope, only using the steroid macro microscope, you can see the PGC. So I think it's a very powerful, very powerful tool to the strong fluorescent PGC. We can isolate that, then continue to doing the analysis. Yeah, it's a very good suggestion. We are further doing this part.
Yeah, I think due to the time, we have to close this section. So let's thank our participants.
Okay.
短时间没有动力。
Ladies and gentlemen, it's time to start our next section. Liu Yiwen from Donghai University, Taiwan, honored to hold the session. Yes. Central Taiwan. Yeah, we have very beautiful campus. Welcome to visit us. Okay, so in this session, we're going to have very interesting talks covering reproduction of economics. Our first speaker of this session is the professor from Department of Life Science, National University of Kaohsiung. So, Dr. Huang, the floor is yours. Let's welcome Dr. Huang.
Thanks for the introduction, Chairman. And I would like to thank Dr. Zhang Jingfang and Dr. Zhu give me this opportunity to open my eyes. Yes, I see I see the name on the paper and embody the really human. Okay, I start my talk. My talk will talk about the variation, ovary, and androgen. Introduction. For the beginning, we have a little biology of angular eel. Eel is the cultured species. The cultured species cannot reproduce in a captivity naturally. So it's quite interesting because in Taiwan we culture many species, and even in Japan they also culture many marine species. But they always a problem on the fish, on the eel reproduction. Okay, for enterprise is a question, but for the academic, the problem is caused by the malfunction of the hypothalamus and the hypophyseal system, because the endocrine system, especially for the GnRH and dopamine, is too strong, and the GnRH is too weak. So in the pituitary, the synthesis of the gonadotropin is totally blocked. So this but in other words, the ovary is ready. The ovary is ready for the exo hormones. So this characteristics make eel is a unique model to study the relation between the exo hormone and the gonadal development. Okay, it's quite interesting because we can put anything you want to see the response of ovary eel. Okay, but beside on the side of the enterprise, they want to more eel. So to solve this problem, artificial propagation of the angular eel existed and to be ameliorated. This means the artificial propagation of the angular eel has been successful in Japan, but the cost is too high. And in Taiwan, we have also success, but it's not announced yet. So it's really a problem. So maybe we have to recruit more the people like you here to enjoy this study. Okay, it's make money. Okay, but for the artificial propaganda, the dilemma is the eel is found in wild population because we cannot reproduce to produce a pure line, and the exo hormone normally we use the pituitary extraction. The pituitary is found in the fish, and the fish is found in wild population. This sector, this reason, like we play the lottery, because each time we don't know the result, and finally we see a result. Okay, and the result will become a bigger black hole. So in Japan, they study for more than 30 years. In Taiwan, we studied for more than 20 years, it's still a bigger black hole. Even the Natural Science Council of Taiwan, they do not support this a bigger hole. Take too much money and no result. Okay, because we play the lottery. Okay. So, but Einstein said insanity is doing the same thing over and over again and expecting different result. We do like this. We always do the same procedure. We want to get the better result. So it's insane, really. Okay. But we try to find a reason why. The reason why is the poor control ground genetics in the animal model contribute to the lack of plausibility. This means if we can understand the genetic background, maybe we can solve this problem. Like now is very popular the precise medicine. Precise medicine, you want to see the genomic background of every individual. Try to treat everyone one by one, not SOP. Okay. And there are many, just like the Dr. Thomas reported this morning. Androgen can reduce the opposite of the primary oocyte. Our study is also on the primary oocyte. And in regular year, the stimulatory effect of the androgen on the ovary development is recognized. This Google Scholar read over 3,000 paper. The keyword is androgen ovary. Okay, so this topic is quite important also. Okay, so we try to resolve the dilemma because we can always play the lottery all the time. So we try to reduce the randomness. The first step is we use the androgen in place of the pituitary extraction. And for the yield, we can do nothing because the yield is from the wild population. But all we can do is traceable because there is a two paper announced that reproducibility and traceability are the two most important aspects of biomedical. So, okay, if we so we try to combination these two to see what will happen. So our aim is understand the correlation between the genetic background and androgen stimulatory effect. But in fact, we want to try to find some biomarker to see before the treatment, is there any biomarker to choose the better responder, right? This may be our goal. So we the material method for the traceability, the yield was operate to take the ovary, and the ovary is put in the liquid nitrogen. The time is 3. Okay, and the yield is implanted with androgens. For do not disturb the yield, the androgen is combined with the tar, the bigger tar. Okay, so we injected the yield 4 weeks, and we have to inject in front. Okay, and in the end, yield are operated or sacrificed to sample ovary patient, and also put in the liquid nitrogen. So we can see the initial state and the final state to see the difference. Okay, we are also use the tool of transcriptome, and everybody knows the transcriptome, so we skip it, and we see the result and the discussion. Okay, we have many years, but we choose three years. We choose in the beginning the ovary. There is no significant significant difference on the oocyte diameter. Okay, so and we choose the yield on this condition and with different final result to compare the cessation from this state to this state, from this state to state to see what will happen. And we try to see the cessation of the genetic. Okay. And although we can see any difference on the mean of egg diameter, but we can see there is a little bit different on the distribution of the egg diameter. So we can see there is a overall markedly this is not significant difference, but for the egg diameter distribution, there is the difference, and the result is here. And we can see the result is there overall there is significant increase, but there is also variation. So the variation of this group is larger than this group. So we try to understand what this state what causes the final state like this, and from this data we maybe have a conclusion. The initial ovary status determine the consequence, the result. Actually, in Japan, they do like this to qualify the brooder, the stock brooder. Okay, so part of our work has been published. You can see the detail. Okay, and in the next step, we investigated the result of transcriptome, and this result is from a commercial company. So it's quite easy. They show me this data and show me this data. Okay, and we can see for the PC analyze, they're really different. Because our topic is want to see the genetic background, so we eliminated the final result. We just keep the beginning state. So we can see for the PCA analysis result, this two group is more close than this two group. So the result of these two is corresponding. So we may say that from the phenotype and the genomic type, that we can tell the difference. But the question is, from the result of commercial company, they give us so many genes. The gene is more than 300 million. Too much, because I don't think this, because the result is the genetic type is result is commons with the phenotype, but I don't think this is good. So we try to cooperate with academia in NICA, and then we remake it, and we mapping the result with the angular yield scaffold genomic, and the number of genes from reduced almost 10 times. Total yield gene is just like 27 million. Okay, so it's easier to manipulate the result, and the result they have a website. If you have interest, you can scan this QR code. And so we have maybe we have the result of more close to the reality. Okay, and we try to find the data, because if the data is sent by the commercial company, you only see the result. You can see what they are done. So, but in this website, you have to calculate by your own self, and we find characteristic of the transcriptome RNA data. Normally the 0.1, the number of biological repeat is low. Okay, less than 10, because it's expensive. Okay, and point two, like the gene expression, gene count are discontinuous and not negative. Right? Okay. And three, the data that does not conform to the normal distribution. Why normal distribution is so important? Because here, what the statistical distribution is suitable for the testing of significant difference is very important. Because our t-test, our star, our different letter is based on the t-test. But if our transcription data is not fit the normal distribution, the tool we use is not work. Okay. So the transcription data is like this. This is the frequency. This is the PFKM. FPKM means the label. Okay. It look like that. In the three group, it look like that. We don't know. Apparently, it's another normal distribution, but it look like Poisson distribution. And that somebody say you like a negative binomial distribution, but more people say it's like a power law distribution. Because the power law distribution make all existing order and the statistical tool invalid. So why? Because there will be large number of extreme element in the power distribution here. From our transcription data, we can see very high level of FPKM, and the more is near zero. Okay, but why? What is distribution? Because the power law distribution is a result action of dependent random variable. The important is dependent. This means what? So the biological mean of the power law distribution is a function like this network. This is we saw family with signal transduction. If the distribution is normal distribution, it's not a random network. Okay, so the biological mean of the power law distribution is in the biological means. But actually, there's a report years ago by a group of Japanese. They said product distribution spectrum from the Gaussian to power law. It did. Depending on your different species and your manipulation, we find the distribution is changeable, not changeable because they are different. But the power law distribution is the core, and the very deviation from this power distribution goal. Okay, so more important is on the other hand, gene encoding the transcription factor and the nucleic acid binding protein must exhibit the power law like intermediate distribution. Okay. So we know that the transcription data is we have three investigation is very important, and we back to the gene. There are many genes like the star, myriad star, as larger numbers of genes. But what can we do? We can do is find their relation, and we will find the constellation of the star. So how to do this concept in our data? We have two strategies. One is cut. To many genes, we have to cut into a small group, like a star. We have a few stars to become a system.
And our method is clustering. There are many methods to clustering, just like proton change, K-means, T-set, UMAP, GMF. Okay, it depends on your purpose. And after the clustering, you have to find their relation, the enrichment analysis. Analysis, we have the KGG pathway or GO analysis. And in this report, I use the KGG pathway. There are 7 catalogs on the KGG pathway. One is metabolism, 2 is genetic information process, 3 is the environmental information process. 3 means signal transduction. 4 means the cellular process. 5 means organism system. 6 human disease. In our analysis, there is no 7. Okay, so we deleted the 7. And the transmission data, we can see that it's like a power law distribution. And we cluster the gene, use GMM into a 50 group. And we cluster and we we do the enrichment analysis. We can see the result. The result is the gene expression is a 2 level, and in many number we have the 3 and 5 subgroup. This means signal transduction and the organism system. If you have a few genes and with high expression level, that means metabolism and genetic information process. Okay, we can see the trend from the expression level and the expression number. Okay, back to our data. We can see this time we clustering the group with 220 group. We can see the different color present different group. We can see in the big high level expression, similar present the phenotype, and the difference is here. There is the difference between the signal transduction and the organism system. And we will continue investigate the data. We can see if we align the gene, we can find that some gene cannot be aligned. This means there is a gene exists in one individual but absent in another. So this make a concept that this means pangenomic. Pangenomic means okay, core pangenomic means the circle outside, and outside there is a section. The intersection there is a core gene, and also beside the core gene there is a shell gene and the cloud gene. Okay, this means there is a good variation between the individual. And if we started accessory, this means if we start, we can see the difference. There is a core and there is a unique gene and there is overlap gene. So this is a core gene, this is a cloud gene, this is a shell gene. And if we study accessory gene unique in one group, and after the KGD pathway, then we can obtain in the section we have 28 pathway. The 20 pathway there is 4 in the group 3 and 19 in the group 5. We can see the scale. So the accessory genes are often involved in the biological process, such as cellular connection, sensory activity, chemical stimulation, and the neurophysiology and other processes. That is very, very important. This pathway is not a classic. It's not classic in the ovarian development. Okay, and the accessory gene can lead a very degree of gene expression difference, which have a certain impact on the phenotype. This means that the variation may be caused by this accessory gene. Okay, after the accessory gene, we want to see the core gene, and with CMM we cluster the core gene, and because there's a number difference. So the core gene we separate it into core 1 and core 2. Core 1 and core 2 by the expression level. And finally we compare the accessory gene, the core 1 and core 2. And we can see that the overlap between accessory and core 1 is little. It means the biological function is very different. And the overlap between the core 1 and core 2 is little. So we can if you look here the KGG pathway, maybe we can make a conclusion. Accessory gene shared or cross gene is a subset the variable gene, and it's generally related to the biological characteristics of chemistry. And the accessory gene set play an important role in genomic evolution and incompetence interact between the genome and the environment. So let's talk about variation. Okay, so in the next, we find that and from our data, when number of genes in each group is aligned, the data contain a large number of zeros. There's many zeros in the before we think it is made artifact. Okay, but it's really existing. The probability of result occurrence is often underestimated the general model. So because there are too many zero values in the data, the same zero value reflects different situations. The counter data often show large variation. This phenomenon is called zero inflection or zero inflection. So because how we find this? Because if we calculate the FC or the change, we have to like this two numbers and we find that it is zero. But we cannot make a zero. But why there is a zero infraction? There is two kind of zero. One called the true zero. It means in the gene there is not expressed. And the other called the false zero or drop out zero. Your technique is meets the data. And there is another term. Zero infraction and EPAV. EPAV means expression presented. It presents an absent variable. It's like a zero infraction. Okay, so in the before we discussing this part, and the next we will discussing this zero infraction. It is existing. So we do the same result. Do the same work, and we can find that the unique gene is very important part in the endocrine system and the nervous system and the sensory system. Well, there's a report means sensory gene always interaction very important. This we make a very okay. Our part of work has been published, and finally conclusion. The genetic background of the ovary is related to the stimulatory effect of androgen. And this pathway includes cellular connection, sensory activity, chemical stimulation, neurophysiology, and other processes. And the strategy to find the biomarker should target on a gene set, not a gene. Yesterday, Dr. Nakamas. Okay. She's a modular. Yes, the sheet modular. It's because because we think 1G is very important, like God. But I think God is the network, not the only one. Okay, thank you for your time. Thank you. Sorry for listening a bit English. Okay. And we have to thank you to the Dr. Lee I mean, our mentality. Thing. Let's see. Because there's support like the data side. Okay. Thank you for this. And our lab group is me and my cat. Okay.
Thank you, Professor Wang, for telling us this very challenging issue of Japanese eel reproduction and your transcriptome approach. Due to the time, we can one quick question. Transcriptome result is going to influence your future strategy of producing Japanese eel.
We enlarge our sample to see the track because we only have 3 samples. Maybe someone criticized it is not enough. There's still a variation in our study. So the future is we should enlarge our sample size and to see what really happened.
Thank you, Professor Wang.
Thank you.
Now it's time for us to move on to our next talk. Our next speaker is Professor Shi Jiahai from Department of Biochemistry, Yongbuli School of Medicine, National University of Sydney. It's yours. Let's welcome Professor Shi.
Okay, thank you very much for a very nice introduction. First of all, I would like to thank Professor Zhang to organize this great conference. I would like to special thanks to Professor Zhu to invite me to this virtual meeting. So today I would like to introduce my research in the antibody translation. Before everything starts, I have a bit of interest. So I'm advisor for comment. Mexico, Brazil Biopharma, which is Brazil providing the body, which is providing the single antibodies discovery service to camel and sharks. Because I basically need this not body targeting yeah, recently and I think everybody know about antibodies. So we know that actually in the war there's two species. There is a heavy chain and white alpaca. So they actually fight IgG antibody groups light chain and become heavy chain only antigen, but we attach. And very few people they actually know that in catalytic fish, shark, and ray, they have another kind of heavy chain only antibody, which derived from the IgM, then the immunoglobulin new antigen receptors. And then in 2019, FDA approved the first nanobody-based medicine actually to boost for the nanobody industry. So based on the prediction that this kind of CAGR for the normal monoclonal antibody, the market is 80%, while for the domain antibody, we believe that this kind of CAGR go up to 23%. And then when I was postdoc, I'm thinking that I actually do some short research. Not many people do this research, maybe I have some chance. And I actually go to see in the literature. It's difficult to write this shot. The first one is another shot is too big. And the both shot is small, but they are very dangerous. You can raise them, you can catch. And the springtail fish, which in the North Sea, they're swimming very fast. It's difficult to catch. And the home shot actually in California, they are kind of small, 1.2, but they still swimming around. Now I'm thinking like that. I search for all the English literature. I can't find any shot for immunization. Now I'm thinking that I'm a Chinese. Chinese usually have two habits. First, we like to eat. Second, we like to farm. Okay, so I'm thinking that okay, so I go to search for the Chinese literature. I use Baidu, I use Fanfa, I use yes, I got something. So 20 years ago, Chinese they like to farm sharks. In this shark, I call it white spotted bamboo shark. They captured the Fujian province, so actually happen a lot in Taiwan as well. So these white spotted bamboo shark, they are very small. The maturation only like 60 to 80 cm, and then they are pretty small, about 5 kg. Usually we wild catch is about 0.5 kg. And then the maturation is shortest time, only 4.5 years or 3 years to 4 years in one time. And then they actually very calm. They usually lie in the bottom of the sea. They don't really swim a lot. They stay there all day and don't move. So it's good for farming. It's small, one person can handle that. Yeah. Most importantly, I found that in 2005, they actually opened a shark farm in Guangdong province, Zhanjiang. And they tried to farm this shark. Unfortunately, because it took 3 years, they actually cannot sell it as a shark in 2008. So it's a very good idea. Unfortunately, I don't have a startup. I don't have money for this shark research, and I don't have track record for any kind of fish research, and I don't have preliminary data. I did actually look for help. So I actually paste, sorry, this is in Chinese, but this is our original format. I paste it like, how do we find support for my nanobody research? I posted it in 2015 May, while I was still a postdoc in Boston. And I look around, ask for help. It's great. I actually, at the beginning, I was originally from Xiamen University, so I can get a connection with those professors who doing shark farming, which published paper 20 years. I get exactly the protocol how to farm shark. And then I don't know where do I catch shark. Actually, luckily I was in City University of Hong Kong. But somehow they have connection in some people in Keelung, this area. So they are able to purchase 20 sharks from Taiwan and capture in the eastern part of Taiwan and then ship from the Taoyuan airport to Hong Kong. Okay, I finally get a shot. I get the farming protocol, and I still don't have money. Okay, and I don't know the shot genome. I finally get the BGI center. They agree to sponsor my research. Importantly, they will do the whole shot genome research. Yeah, so they spend a lot of money to do the intensive genome research. The short genome size is around 300 million base pair. They sequence 100 times. They actually get around 30 billion read. So we actually assemble the entire genome to around 51 chromosomes. And the important part is, for me, I'm actually I'm particularly interested whether they have this immunoglobulin new antigen receptor. Yeah, they have in the chromosome. They actually show that they have the MHC-based immunity, which indicate that they might have antigen-specific immune response. This is, at least to my knowledge, is a very comprehensive bamboo shot genome method. So it allows me to do, we know the exactly sequence size, sequence antigen. But and then we do a comparison. I would say that I'm just lucky. I'm just lucky. Because catalytic fish separate from bony fish around 450 million years, 450 to 480 million years. So they have a wide spectrum of catalytic fish. Elephant shot, cat shot, wine shot, whale shot, and then lucky. I'm just lucky. I choose the bamboo shot. They species. Well, different from elephant shot. Elephant shot, they will come by the IgA army, but they may not really have this kind of antibody. And for cat shot, they actually have immunoglobulin new antigen receptor genome, but they don't have antigen-specific immune response. It just for our shot, they are really late development, so they have this antigen-specific response. Well, we are lucky or unlucky, because there's another group in Japan that should publish a genome sequence paper just half a year earlier. So they publish in Nature journals, the Nature sister journals. And this sequence still not precise enough, because we need to know the detailed base pair. We finally find out that we have some of the genome. So we can see that in the short genome, they have IgM, IgNR, they actually have IgW. But this genome sequencing, although we have done a lot of data on it, but they are not precise enough. We cannot find the IgW. We can only find IgM, and then we find IgNR, particularly located in the chromosome 44. Yeah, we find around 7 IgNR plus 37 IgM. And then we actually combine the second generation and third generation to sequence the detailed sequence of this 2 megabase genomic region to get a precise sequence. We finally get the result is here. It's very interesting. So we have 7 locus, 1, 2, 3, 4, 5, 6, but only 4 of them they are complete. And this gives us great surprise that the short IgNA they actually sequence actually are similar as other bamboo shoots. They only have very limited recombination. This only gives by 7 possible shots. Yeah. Although we have other locus, but we can see in the later research, only this part will give him response. Other part is verified. And then we do the whole body transcriptomic analysis. We found that in the different tissue in the blood, we can say they actually have 3 IgA locus expressed. Brain, eye, grill, liver, spleen, they actually have a lot of IgA expression. Pancreas, and the spiral bar. Spiral bar is a specific immune organ. And then we also get this down the info. We find that actually IgA1 specific expression down, and IgA2 ISOCCE actually. So now we know IgA exists in the genome locus in the mRNA. Now we want to see the really as a protein. So we get the plasma protein, we do the Western blot. We find that yeah, they do have this IgA monomer. And then we in the reducing gel, we remove the disulfide form, we see a very heavy band corresponding to the weight. And then we do a really long running native gel. We find that actually exists as a 3 different oligotrimer, tetramer, and actually. So we finally know that we have IgA. Now we want to do immunization. We actually in Hong Kong we assemble a very simple culture system, not as fancy as Professor Zhang's institute. Really fancy fiction. We are very actually buy everything from Taobao, so it's very cheap. But luckily this side is very happy because it even let eggs in my culture system. So suggest that these drugs are very healthy, and then the maximum time they can stay in this town for 2 years. So they are very happy in this system. We perform the 4 different immunization protocols. We either immunize them weekly, bi-weekly, or monthly. This is injection site at the underwing. And then we also do this is intravenous, this is IV injection to see whether they are working better or not. We use this antigen, so this is an infrared fluorescent protein, 1 to 3, and then this is link protein. We show that actually the titer increase over time. So the best one is immunization actually in the high ISO. So both antigens they actually have the small, and we can see in the Western blot they actually increase over time. And then we do the sequencing. We try to see where this IgA sequence comes from. You can see based on the different shots, majority of this sequence, this antibody, are mainly come from the IgA1, not 2, 5, 6. Yeah, you can see in the frequency of the gene usage, they're always IgA. And then it's interesting here, and then we see the variation of this antibody. Find out that the CDR1, so different from traditional antibody. In shock antibody, they only have CDR1, hypoviable region 2, hypoviable region 4, and the CDR3. So you can see interesting here is the CDR1, H3, 2, and H3, 4, they don't really change much. The geodiversity comes from the CDR. The CDR3 is longer than the traditional antibody. It actually has a big size, around nearly 18 amino acids, and then up to 40 amino acids. So it's larger than the traditional antibody. Suggesting that is why only the CDR3 variation is enough to provide this binding effect. And then we actually try to do the fish display to screen out what kind of nanobody actually have this nice binding affinity. We're actually getting 7 nanobody that they actually found pretty good. The green fluorescent protein, we get the highest KD go to the sub-nanomolar binding range. And this is not the end because we are doing domain antibody, which is different from traditional antibody that we are able to do antibody engineering. So the good side is that this is the pure physics story. If we are able to engineer 2 domain binding protein for one particular antigen, if these 2 bindings they are independent from each other, if we link them with covalently linked like the classical, theoretically we are able to significantly increase the binding affinity between this nanobody to this antibody. So therefore, we actually choose our nanobody to pair up them together we actually generate one super nanobody. So the binding affinity go up to 20 picomolar, 27 or 20 picomolar. Measured by the vehicle, it's a very small binding. So based on the literature, this is the highest GLP binding nanobody. But there's another company claim that they can generate another VHH binding there, so you have nanobody binding together, they can use one picomolar binding affinity. So really, it's like one picomolar really the current detection technology which is a terminal power of Milo 15. Yeah. The vehicle and this machine which is so therefore our data suggests that we prove that bamboo shark can have genome locus for IgNAR, and they actually have IgNAR molecule in the bloodstream. And then this bamboo shark indeed can process the human antigen-derived IgNAR immune response. And then use this bamboo shark as a model, we are able to screen out high binding affinity, high specificity nanobody. So this is a very brief table messaging is that why small bamboo shark the serve as a model system to stimulate 3 to 6 months, we can generate high affinity shark nanobody. Additional nanobody can serve as an intrabody and cellular target. We can also do the antibody engineering to reach picomolar binding affinity. Currently, our lab is trying to prove that we are able to generate naive shark nanobody. We can also generate high affinity shark nanobody. And then this is all published work. So my lab is continue to understand the mechanism of shark can generate high affinity. After this one, we try to first do this kind of whole body genome sequencing here, whole body transcriptional analysis. We're taking the 3 adult shark, 3 junior shark with 1 male and 2 female. And the adult shark are all female. This is by chance. We take the 20 tissue out from here. Unfortunately, we didn't take any bone because we actually don't know about that. Yes. So we take around 20 tissues. We collect more than 15 billion read. We assemble all of them. We identify for this shark they have 15,000 mRNA plus around 44,000 isoform. They also identify around 3,000 non-coding RNA with 3,000 isoform. And then we actually find based on the different tissue. So this is the mRNA we identify from the different tissue. And then in the flow model, actually we can see that this is coverage is pretty good. They coverage most of them, but we don't know which one is successful. I don't have that kind of knowledge to identify that coverage. And then we actually compare. We do the comparison from other animals. We find that very interesting. Catalytic fish is similar, they evolve by themselves. They only share the 10% of the gene conserved with other species like human, mouse, camel, and other fish. And the catalytic fish itself, it actually evolved by itself. It actually similar as we show that the bamboo shark is the highest one, homology to this bamboo shark. But for cat shark, whale shark, they are kind of less with bamboo shark. And the elephant shark, which is much earlier elephant shark, they don't even have an IgNAR. So they actually share the minimal homology with bamboo shark. And then we take out this gene to see what are these genes. Interestingly, we find that these genes are actually enriched for immune-related genes. So this data will probably explain that why bamboo shark will have antigen-specific immune response. Maybe they have a certain genes different from other shark, particularly they may even not conserve with other species. Yeah, and unfortunately, the student in my lab doing this kind of research want to venture capital. Is I'm not. So this one still stop there. So finally, I actually don't know there's many people study fish spongenesis. So I actually do some kind of conservation of sharks because we know that biology. A lot of my friends they actually do environmental conservation. I want to do that. I'm working on shark, but I also do the shark reproduction. So I do the shark. As my student who actually correct the shark sperm. So first time you show me this. Are you joking? I study biology for more than 20 years. Do you want to show me this is a sperm? I'm not stupid. My student saying that. Okay, I don't go back to do more. So finally, I agree with him. I actually talked to him and said, yes, I think that I'm stupid. So we do a DNA scan. This is a DNA. It's the first time I see a DNA could have this strip, spiral strip. This is repeat. And then this is and then my student even show me a video. Okay. It's a bi-directional motion of the sperm. You can go back. Well, you will see the neighbor again. So this is the tail disappear. Only the head. Head still can move. Maybe so we just see this again. So the actually go back. Turn right and then stop a little bit, turn left. So at that time we are very poor. As you know, all know that I'm very poor. I don't get any research money. So I have no money to buy the CASA system to analyze the sperm. So I try to persuade my friend to mechanical engineer. With mechanical, he's a mechanical engineer with train Japan. So he's able to do the data analysis to see how the movement of this sperm. He showed me the sperm movement, the head actually contribute to 30% of the driving force. The tail contains around 70%. The head is 30%. But if in the kind of backward motion, the head contributes 100% of the. So therefore, my friend actually designed a robot. Our robot is 3 robots. Only the head, tail, and the head and tail together. The first one is the head and the tail together movement together. The actual movement is the fastest one of all these swimming stars. So this is very surprise. So we finally published a paper after 3 years, battling with the. So finally, I would like to thank. There's a lot of people to help me. So because, for example, the Stanford University, the professor Su Yuanquan actually showed me all the protocols. Leon Chang from CTU, so he helped me to get all the shots. So he was share one student for me. And the Dr. Sun actually do all the mechanical part and the sperm movement analysis. And then Dr. Yang from BGS and then do all the sequencing. I particularly like to thank the ocean part. So they actually help me to handle all these sharks and handle the shark sperm. And yeah, I would like now before I actually get the grant from Central government to support this research. And actually, I used to work in Hong Kong, City University of Hong Kong. Later, I moved to Singapore. Yeah. Thank you very much. Welcome.
Thank you, Professor Shi, for this exciting talk about bamboo shark. Okay. Any question from the audience?
Similar as all other species like the mouse or rabbit. And then the critical part of this one, I can produce this antibody in E. coli. So basically, the amount of the antibody is unlimited. I can use fermentation. That's scale up. So I only need to take 1 ml from this shark, and then I will do the fish display, and then I get a sequence. I produce this.
That's again another curiosity. So they have adaptive immunity. And so developmentally, during what stage can they start to make an antibody? When they're in the egg case, they start to make it.
Okay, to answer your question is traditionally we believe in the embryonic stage they don't really generate antigen specific immune response. But however, for other people they use nurse drug. They can only handle nurse drug in their younger stage. They can so for nurse drug is able to generate antigen specific immune response in the junior stage. Okay. But as my data showing here, in our bamboo shot is they actually in the junior stage the preference is actually R6. And only in the adult stage is actually R1. Okay, we can see about that. So I assume that I didn't do the junior stage the bamboo shot. So I assume that usually at the junior stage for this antigen specific response may not be that super. May actually cost more time to
just is this possible to for you already mentioned the environmental application or medicine application for the this type of strong affinity binding with the
Yeah, they actually have a few companies setting up for that. For example, University of Aberdeen in the UK, they actually have a small program for the shark nanobody program. They actually use shark nanobody to against breast cancer treatment. In Australia, there's around 3 to 4 companies to actually develop a shark nanobody for different purpose. And in the US, I don't remember actually. But the company I have in Hong Kong is not competing with this kind of established company. So we are not going to do drug development against cancer, but we're doing for research reagents, for example, for antibody or resistant antibody. Because the beauty of shark nanobody here, shark has a very high urea in the bloodstream, or 500 millimole. So this indicating that the early shark plasma protein have a very high stability. So for nanobody here, shark nanobody have a almost stability in 80 degrees Celsius. So basically, when we want to do Western blot, we don't need cold chain anymore. We can just store the antibody in the room temperature. Yeah. So this is the beauty of the shark nanobody. And then there's another beauty of shark nanobody here, but because I really didn't detail here is you can see here, the antibody, shark nanobody, different from any other antibody is we don't have CDR1, CDR2. So our CDR3 is very low. Okay, so it may actually benefit for both GPCR drug design. Because GPCR drug design, the cavity is very deep inside. It actually allows root to go deep inside. So there is a drug design strategies. They use a very long CDR3 to deep inside the cavity of GPCR, and then we just cutting out the CDR3 as a small peptide drug for therapeutic purpose. But this one is a many people is trying to develop. Yeah, long CDR3 based strategy drug design and the Pfizer in the UK there. So that's why in the entire Europe, these big pharma companies, they always have this kind of shark nanobody program. Yes, so for this sperm, so for the tail, they are very similar to traditional sperm. Right. No, yeah. The head, you're asking the exactly same question as the reviewer. Okay, so we try to fly to see the head. Okay, where the machinery, how you turn the head change. So the tricky part here is although actually we can see here, they have a mid piece here. Okay, and then we can see usually for this one, rotation is turned to the left. Okay, and then the inside, they got typical 9 plus this kind of 5 point, very typical motion. However, for this location, they locate faster. In terms of speed, they are 3 times faster than the tail. Okay, 3 times faster, but their wave is smaller. But the wave actually is smaller. So they actually get 3 times faster. But every time we try to do the cryo EM, right? This part works. So answer your question, we don't know yet. We actually don't know yet. And now because I actually move out, I actually cannot get the people to correct the spell okay.
Can I ask one more question? Can I ask one more question? That's amazing when you mentioned that for GPCR, because pretty much all the GPCR antibodies for the smaller GPCR, they're all junk, all the commercial. Even papers published in Nature, they are junk. Those data, the Western blot data are junk. The antibodies are not working. So I wonder how easy it is to get antibodies for the smaller GPCRs with small extracellular portions to generate very good, authentic GPCR.
You ask you very important question. Very important question. We have tried. Okay, so we have so my friend in Shanghai actually have tried to working on the GPCR antibody. Okay. Usually we are successful experience for GPCR which have a larger extracellular domain. Yeah, that was fine. For small GPCR, we all fail. We all fail. We actually build a very beautiful. My friend actually built a very beautiful system. So we don't actually trust the traditional face display or display, surface display to screen out the nanobody against GPCR. Actually, my friend actually developed a mammalian cell surface-based screening system. So they actually base a 3D intelligence integrate, actually try to mimic the technology in our human B cell maturation. So B cell maturation, they actually mimic over cell surface receptor and to against another cell express GPCR. Okay, and so we express GPCR on 293 T cells. And for small GPCR, really difficult because their extracellular domain, they somehow will interact with other glycoprotein or whatever stuff. They don't really expose. We don't have good successful experience for that. We have successful experience for other membrane, not for small GPCR.
Thank you.
Great.
In the audience for so many interesting questions. Thank you, Professor Shi. Now it's a bit delayed, so we have to move to our last talk of this session. Last speaker of this session is Professor Li Guanzhong from Department of Agriculture, National Taiwan Ocean University. Switching our focus from Ben Bouchard to Big Thing Sweet.
I know all waiting for lunch. I will go fast. Okay. So my talk is about the cephalotes. But before I introduce my new study, I would like to say, well, just beginning, as you know, I graduated from Dr. Jing Feng's lab. So we have a lot of collaboration in think chain, living fish. So today I will not go to start. So I will talk about the communication between the squid and its life profile in the necessary female organ cephalotes. So I think most of the audience here should not understand what is the necessary female organs. So here I just show you the structure. So you can see here is ovary, it is orbita. So they have three different accessory organs. It's a NG, implemental gland, accessory implemental gland, oviductal gland. So these three glands, their function is to formate the egg capsule of eggs. Okay. So this slide just show you the difference in the microbiome and in different hosts. So you can see in uptail suite, they only have one symbiote. But if you check with the ANG, this data has come from our lab. They have more than 500 microbiome in their ANG. So this is the reason the live organ can the most well-known model to study the host and the bacterial infection. And then nobody tried to know the ANG's storage. So they have too much material inside. And as we know, we can see a lot of different well-known models to study the interaction between the host and the maybe the algae, fungus, and the bacteria. But they are non-cyclical symbiosis. It means it's hard. You only can understand the host and microbiome communication, but it's hard to know that any correlations with others. So in here we just show you a case in uptail suite. So they have a cyclical symbiosis. One is a daily cycle for live organs. Another is a reproductive cycle in NG. Okay, so now we have to show you some interesting part of my studies. So here you can see they have one more organ called live organ that attached on the inside. So this live organ, they don't show the sex difference. It means male and female, these organs. So the NG and NG is female sex. So in here you can see through the different species like squid, lollipold squid, cuttlefish, and the oxley. The live organs only observed in lollipold squid and bottom squid. And the NG only observed in lollipold squid, cuttlefish, and oxley. So in our case, we choose the oxley. So, but now we have a question, right? Why we choose big fin reef squid as our model to study the host micro interaction? So this is a picture I took in the fish market near our university. So my reason is that only one species, big fin reef squid, can be found in Taiwan. So this picture I took in Malaysia. So could you imagine, or you can identify how many different species in this picture? For me, I can identify at least 3 different species. The first one is cuttlefish. Number 2, you can see the large size, smaller size here. Okay, this is the rolling squid. Then 3, almost. However, if you check the genetic background, this part, it should be more than 4 species here. So this is the reason why I did not choose the logical squid as my model. It's because too complex. Every single individual, I need to check their genetic background, which species. So how about the function of the live organ and NG? So according to the study from other labs, they say this live organ hypothesis is for prevent the adapter to hunting them. Okay? And the ANG, they have okay, here is just a picture showing you here. So you can see the ANG, NG, and OG, they are the component or egg capsules. So the ANG also deliver the bacteria into the egg capsules. Okay. So what is the function of this bacteria? Okay, so this bacteria they can prevent the fungal flooring in the egg capsule. This means if you treat with antibiotics to kill the bacteria, so this egg capsule they will not immediate, they will fast the bioflooring cover the shell. So our first thing is to check this ANG bacteria. Do they show the selection by host? So according my cooperate with Dr. Yang Sanhua in National Taiwan University. So according to the sentence they show in the paper, he said also the bacteria consortia of ANG show the variety different suburbs. Its composition shift gradually during female maturation. It means this ANG bacteria they selected and by the host. So we have our first goal is to understand the bacterial selection mechanism during female maturations because, as you know, several have an optic gland, the organ have a similar function as a pituitary, and it should have some endpoint communication between optic gland, ovary, and ANG. But today, we're not just beginning for this studies, so we have to discuss this area. So the first thing is to understand the host selection. Okay. So before we go this first, we have to check this selection they correlate with female reproductive cycle. So first we have to determine the ovarian stage. So here we just show you the histology for the oocyte. So you can see the oocyte from the early stage and to the large size. Okay. So we check the heterogenic expression. Okay, I would like to mention this heterogenic expressed in follicle cell. So the follicle cell is we can see they will embed to the oocyte. So we can check the egg one by one. So here just show you the VTG expression. So based on this, we identify 4 different states. It's a juvenile female, they have a primary oocyte. And mature female, they have a multi-layer follicle oocyte, heterogenic oocyte. And maturing female is an early stage of heterogenic oocyte and mature female. Okay. Okay, so here just show you the relationship between the female from female maturation and the ANG development. So you can see in mature female, the ANG only have a small set of the colony inside. So relate maturing, you can see they have more colony inside of ANG. And in mature stage, they will show the pigmentation ANG. So we have a hypothesis if they have a universal and specific selection of bacteria during the ANG growth. Because during the early phase of the ANG formation, you can see they have a lot of outer layer of the epithelial cell. And you can see they have like a pore, or they can migrate from the outside to the inside. So we call the early stage is a transmission, and then they colonize in the ANG. So we call the early stage is a universal selection, and the late stage is specific selection. So we took two genes to test our hypothesis. So first is transferrin-like protein. So in other modes, the common antimicrobial mechanism innate immunity based on the development of transferrin-like protein. So we want to know the PF expression patterns in NG. And the other is a specific selection by peptide or glycan recognition protein. So in vertebrate, the innate immune system include this protein to recognize the microorganism. Okay, so first, here we just show you the in situ hybridization data, so you can see the PF gene is expressed mainly on the outer layer, AG. And the least expression, according to ISOCCE staining, is very stable. It's very stable during the NG growth. We can see that in every stage they are mainly expressed in the outer layer of NG. And you can see here, you did not see any signals of PF observed in the inside of it. So we move to the PGRP. So according to transcriptomic data in ANG, so we found 4 different PGRP in 15 leaf squid. Okay. You can see they have different characteristics like membrane form and releasing form. So here I only show you the PGRP2, 4, and 5. It's because the PGRP3 only expressed the humus. So here the this PGRP2, 4, and 5, you only see the PGRP2 have significant difference in age for 2 female and 2 female. And the PGRP4 and PGRP5 they show significantly reduced. So we choose the PGRP2 for next experiment. So as I said, we said it has the outer layer and the inner layer. So first we like to check this PGRP expressed in the outer layer or inner layer. So here is the histology. NG, so you can see that the outer layer and the transition area and the inner area. So you can see the PGRP2 highly expressed in the inner layer, and the PGRP4 and PGRP5 higher expressed in the outer layer. And we can see the PGRP2 have significantly increased in pure stage. Here is the most interesting thing in PGRP2 expression because you can see here it is a situ hybridization result. You can see the PGRP2 expressed in the inner layer is not stable. So it means they are expressed in some tubular, but they offset in another tubulars. And we isolate the different colors of the tubular. So we analyze this PGRP2's expression, you can see that show the very different expression levels. However, if you check the tubular color, okay, unfortunately, they are not correlated with the color, tubular color. They may correlate with another something I don't know. Okay, so at least based on the PGRP2's expression and the transferase expression, we may think about the NG have a different regulation in outer layer and inner layer. So we move to the transcriptomic study. So in here, just show you a global expression pattern. So you can see that in early stage, stage 1, stage 3 is the transmission phase and colonization phase. They are grouped clustered in the same cluster, and the mature one is another cluster. And we later to check the expression pattern of the gene, so we can see that 5 different expression patterns were observed DG. The first G1 is they highly expressed in the stage 4, and the G4 they are significantly reduced in stage 4. Surprising. So this one means they are highly expressed in stage 4. The most gene is for the lipid metabolism and the flora fermentation. And in G4, is the most gene, surprising, most gene is immune response in apoptosis. This is a little bit weird for our knowledge because as we know, the host will try to use the immune system to regulate the microbiome composition. But in here we showed that when they had more bacteria, but they reduce the expression of most immune response gene. So the interesting point from our study is most immune-related gene had reduced transcriptomic levels. And in the last stage 4, mature one, they have lipid metabolism, and the flora fermentation is very important to regulation of bacterial pigmentation. So I'd like to thank Dr. Chang for everything support, and Dr. Zheng for raise the screen, and Dr. Yang for bacteria analysis, and Dr. Wang to help us to analyze, to identify the different species of developments. And Howard Lee for most analysis and histology, and the Hong Wei Zheng is for bioinformatics analysis. And thank you for your attention.
Thank you, Professor, for your interesting talk. Any questions from the audience?
Thank you for your talk. I wanted to mention to you there has been quite a lot of work done on the bacteria of the developmental glands in cephalopods by the team of Renata Rocher at the museum in Paris. And among the interesting protocols they had, they developed co-culture between cells from the cephalopod and the different strain of bacteria. They have been isolated, and it's some bacteria really are able to grow and survive only in this co-culture. So this kind of protocol could be interesting for you to further investigate the relationships between the bacteria.
Yes, I think it's very important for our study because we already tried to clone the bacteria in ONG, but at the least maybe more than few hundreds. So we will check clone one by one, and most of the clone is not grown to the major group.
And that's following the same question. They started to clone bacteria. The purpose was to isolate antibiotics, for instance, because, as you said, they protect the egg against other bacteria. And some bacteria they could only cultivate with cell culture of the host. So it's something.
Protect the egg shell, right?
Yes.
So what do we know about the bacteria in terms of controlling development of the egg?
In other studies, in bacterial, if they treat with antibiotics in early stage, it means they not yet transfer the bacteria inside of the ANG. They will reduce the ANG size, and even some of the screen will lose the ANG. So they hypothesis that some bacteria will stimulate the ANG formation.
I'm just curious about the ovarian contribution of the bacterial growth. So if you were to take ovarian extracts and then inject that into a juvenile, would you see the similar growth of bacteria?
We try to do some treatment in squid, in whole squid, but unfortunately, when we treat with chemicals by injection, all the squid will die.
No further question. This wonderful talk. And it's almost 1 p.m. and we have to close this session in.
We have simple lunch as you wish for 30, 40 minutes, and then we will depart from here to our trip. Yes, thank you. Welcome everyone, including both. Welcome.